All right, I'd like to thank the organizers for having me. I'm going to tell you about some of the work we're doing in my lab. I'm very interested in this problem. I think a lot of us, it's come up in some of the talks. This question of how the building blocks of life encode for information on larger length scales. So you think about proteins and RNA, their interactions happening down here at the nanometer length scale. Somehow those interactions are encoding for information on how to build structures inside the cell. And then, the biophysical properties, composition, size, et cetera, of these structures is then going to dictate decisions that impact even larger length scales, cell growth, differentiation, and so forth. And so this is a really deep question. We've been thinking a fair amount about, how do you understand from a fundamental physics perspective the links between these length scales that encode for information on these larger length scales. Okay. As physicists, we know that there's this really beautiful framework for thinking about one way in which molecules and the interactions on really local length scales can propagate over large length scales, and that's the idea, the formalism of phase transitions. We think about dilute disordered states and small molecules like water in a gaseous state condensing into more concentrated but still disordered liquid-like states, and then crystallizing into solids. And this is the kind of stuff we see in winter, or if you hot shower or something, condensation on the mirror. These macroscopic visible structures that form from the angstrom level interactions can really encode for information on these large length scales. So this is the kind of thing, we don't imagine that that we can have a formalism that explains everything about life, this sort of new physics that emerges, we think, but we like to, we're inspired by things like this, right? That really powerful framework for understanding assembly. Okay, so just thinking about this problem of assembly of structures in cells, sort of this intermediate length scale in the slide I showed at the beginning. This is structures inside cells. When we think about what's going on inside cells, we're often thinking about these organelles. Usually what comes to mind is little vesicle-like things, right? Little membrane-bound bags in which reactions take place. And that's nice because it compartmentalizes the interior of the cell and concentrates reactants and so forth. What's interesting is that there's a whole other class of organelles which are not membrane-bound vesicle-like structures, but they're really just dynamic assemblies. They typically have both RNA and protein, and they go by various names like RNA protein bodies, RNP bodies, RNA granules, or nuclear bodies, and in the nucleus. So here's some examples. I'll just show you some, just very briefly to get started. Let's think about the germline P granules in the CLD embryo, processing bodies, and essentially all of our cells are these structures that are involved in mRNA turnover and decay. And then in the nucleus, there's dozens of different types of these RNA protein bodies, which are involved in a whole bunch of RNA processing and regulating the flow of genetic information from DNA to RNA and protein. And despite the ubiquitous and important for all this biology, there's really not been much at all understood about how they assemble sort of mechanistically from a fundamental physical perspective. So over the last few years, we've been spearheading this idea that these structures represent condensed liquid phases of the cytoplasm or nucleoplasm of cells. So these are these germline P granules, and we started thinking about these and how they are segregated to one half of the embryo. And in the course of these studies, we found that if you apply these P granules inside a syncytial gonad, you can apply shear stresses in these structures, drip and flow and de-wet from the surface, and all the classic liquid behaviors that one sees with things like oil and water. This led to a model, and I'm not going to go into any detail, just suffice to say that a bunch of data led to this model where the anterior-posterior axis of the embryo spans this two-phase boundary such that these droplets dissolve in the anterior and condense in the posterior. We went on to show that these, this liquid-like behavior, dense liquid phase behavior of RNA proteins, not just these P rings, but nucleoli also have liquid-like features. These are the frog system, which is essentially an emulsion of these droplets, and you push two of these nucleoli together, and they fuse around it into a single larger structure. More recently, we showed that this emulsion of these droplets has to be stabilized by a cytoskeletal network. It's actually really interesting because it's an actin network inside the nucleus, and if you get rid of it, all these droplets coalesce into a single larger droplet, and they actually do it by gravitational sedimentation, which is really weird and interesting. I'm happy to tell you about the physics there. We've started to get some handles on the molecular players involved in promoting these phase transitions in cells. A lot of this has to do with disordered protein motifs, so not compact, well-folded proteins like we're used to thinking about. These are really disordered polymeric chains. In vitro, they form beautiful droplets, and you can express these things in cells and see structures very similar to the organelles that we see. And we've done some microbiology approaches, and you can measure the viscosity and viscoelasticity of these things. This is, I would say, still a working hypothesis, but it's, the evidence is really mounting that there's a kind of liquid-liquid phase separation that happens inside cells that underlies the assembly of many of these structures. So we've become interested in various aspects of this problem. We're trying to understand what are the molecular players that govern the assembly and how they connect to the biological function. One of the things I want to tell you about today, which is kind of one of the perspectives we've been taking on this, is this question of how these structures, these liquid-like organelles, are related to cell growth and size control. So the assembly properties and function of these things, how is that connected? Is it connected to the growth and size of cells? And this maybe it seems like an out of the blue sort of question. Why are you asking this question? And part of the origin of this came from thinking about one of the structures that I've already showed you. I've shown you a few pictures and these fusing droplet-like behavior of nucleoli. So nucleoli are sort of the nucleolus, right? It's the structure inside the nucleus. And the nomenclature is kind of unfortunate, but so these are nucleoli in tissue culture cells. And if you've ever looked at HeLa cells or any other NHP cells or anything, you've seen these things. They've been identified for 150, 200 years actually. And so they assemble, they're RNA protein bodies that assemble around sites of nascent ribosomal RNA transcription inside the nucleus. And this is important for cell growth. If you think about the central dogma, the software of the cell, the DNA, encoding the hardware of the cell, like. So if the cell is going to grow, it needs to make a whole bunch of protein. Most of the dry mass of the cell is protein. And to do that, it needs ribosomes. And that's really what the nucleolus does. The nucleolus is making the ribosomes, and that's the machine that makes the hardware of the cell. So if the cell is going to grow, it needs lots of ribosomes to pump out a lot of protein. And because of this connection, it's well known in many model organisms and many cells and so forth that mutations that affect ribosome biogenesis and nuclear assembly have an impact on cell growth and size control. This is just one gene that gives basically the phenotype is large nucleoli that are hyperactive, spitting out lots of ribosomal RNA, this rRNA. And then the cells are bigger, and these are C. elegans worms. The actual whole worm is even bigger. So there's a lot of examples of that, cells being larger than normal, or in some cases smaller than normal. Cell size and growth phenotypes associated with the nucleolus and ribosome biogenesis. And so that's why we started thinking about the connection between these droplet structures and cell size. Okay, so we started looking at this and using the C. elegans model system to probe, sort of biophysical level, to probe what is the link between the assembly of these structures and cell size. So this is a C. elegans embryo. You can see these nucleoli. You see two nucleoli will fuse into one. And we had a nice handle in this system because the embryo in the wild type, normal normal C. elegans worm comes out as 50 microns long. But we knew about a few different RNA interference conditions where we could make the embryo either smaller or make it bigger. And then we asked the question, what happens to the nucleolus under these conditions? You make the embryo big, all the cells get bigger. What happens to the nucleolus, or vice versa, with making everything smaller? So this is the experiment. We can change the sizes of the embryo and the cells pretty significantly. And so the question is, what happens to nucleoli? Do they get smaller or bigger? So in the spirit of audience participation, I like Paul Selden's talk, and I also have a beard. Do nucleoli get bigger or smaller? So who thinks bigger? Sorry? You can do this with your eyes open. Who thinks they get bigger? Okay, who thinks they get smaller? Who thinks nothing changes? Okay, good. So we had quite a mix. I thought, okay, that's good. Well, it turns out with this experiment, and nucleoli get smaller in bigger cells. So these are the big embryos with the big cells, and they have small nucleoli. These are the small embryos, small cells with big nucleoli. So essentially, that's weird, right? Although many of you guessed that, so that's good. Pat yourself on the back. So what's going on here? What is the origin of this? This is actually very different than what's seen with most organelles, most structures in cells. Actually, this is called size scaling. You make the cells bigger, and then the structures get bigger. That's seen, for example, with spindle and centrosome and other structures. We started to realize what was going on in this system, and this is why we had a spectrum of answers, because it depends. And if we're thinking about this as maybe this is a kind of phase transition, you can say, well, what's the concentration when I'm doing this, right? What happens to all the components? In this particular system, for detail, for reasons that I won't get into that have to do with this worm anatomy, this is essentially how the oocytes are produced, how the embryos are produced. You're loading a set number of molecules into the embryo. And because of that, so if I put N molecules into this box, and then what my RNA conditions are doing are basically changing the size of the box. So if I make the box really big, the concentration is low. If I make the box really small, the concentration is high. And this is just a very simple model we built to try to quantify this. It's basically, we say, okay, there's some number of molecules that are loaded. We've shown that that's constant under all these conditions. And we change the concentration. If you make the cells really big, the concentration is low. And if there were some kind of phase equilibrium between the condensed phase and the dilute phase, then eventually you hit some saturation concentration. If the concentration drops to some level, you'd essentially dissolve this thing and wouldn't even see them anymore. So as the cells get bigger, the structures get smaller, actually until they dissolve. So this is just a very simple model that shows that. So the size of the structure, the integrated intensity, because there's some expanded fraction when we measure it that way, is proportional to the degree of supersaturation of these nuclear components. And this model is very simple. It has very few parameters. We can actually sort of independently measure these parameters from a few different other experiments. This is the data. This is the way we plot this is max nuclear intensity. Again, since they spin the fraction, we're using integrated intensity of these structures. And we plot against nuclear volume. We could just as well plot against cell volume, though. And so you have this inverse relation: big cells, kind of small nucleoli. And this model does a pretty reasonable job with no free parameters. The data is a fair amount of spread the data, and maybe the slope's a little off, but with no free parameters, I was quite happy with that. It's actually not only qualitatively gives you the right answer, but it goes pretty much straight through the data. Okay, so we started using this model and this experimental system to try to map out the phase diagram, a preliminary phase diagram of this structure. And so this is some kind of interaction parameter which we don't understand at all, so this is sort of a cartoon really. As a function of concentration, this is a variable we can really control it. As you make the concentration higher and higher by making the embryo smaller and smaller, we get deeper into this two-phase region and the nucleoli get larger and larger. There's an interesting prediction that came from these experiments is that we noticed all of this data that we took was from the 8 to 32 cell stage of the embryo. If you look at the very early stages of the embryo, the nucleoli basically don't assemble. So there's something developmentally that seems to control the interactions that govern the assembly. And so you see in some of these elastomers, you see really small transient nucleoli, other ones nothing assembles at all. And so we hypothesize that what this means is that there's some developmental control of this interaction parameter pi, such that maybe you're up here, so at the wild type concentration, nothing assembles. And so we tested this. We looked, in these early embryos. If you so here's the wild type one, wild type concentration. If we make the embryos larger, concentration goes down, nothing happens. But if we make the embryos smaller, and in these early cell stages, we induce nuclear assembly. So it looks like you're basically driving assembly by jacking up the concentration above a new saturated concentration. This is somehow developmentally controlled. Okay, so I'm keeping an eye on the time. I've been warned, so I think we're doing fine on time. Okay. So many of you may be asking at this point, everything I've said is, this equilibrium concept. It's fine, maybe a test tube or something, but equilibrium in a living cell, isn't equilibrium death, right? And so that's true. Life is intrinsically out of equilibrium. There's all this, ATP-dependent stuff happening and changing, concentration changing. And so this is something we've been thinking a lot about. And I'll give you, and actually, we've been thinking about this for a while, and some of the examples I showed you before, with the p-granule story, there's a non-equilibrium feature in that the saturation concentration varies across the embryo, and I can tell you more about that. And also with these nucleoli, we've shown that when you deplete ATP, then the apparent viscosity goes up by about an order of magnitude. So there's already hints that there's non-equilibrium features. Maybe we can start to think about these things as active liquid droplets, okay? And I'm going to tell you about some work we've been doing to extend the simple model that I just showed you in this early embryo, and it's starting to give us some insights into non-equilibrium features that are at play with respect to this nuclear assembly process. Okay, so I showed you, still images of nucleoli, and I showed you, it's nice. You saw these two bodies that form, and they form around these specific gene loci. But if you actually look at a movie of this assembly in time, this is an embryo, and I'll play this movie, it'll move through. As the cells go through the cell cycle and divide, and the nuclear envelope reforms, and the concentration of components increases as everything's loaded into the nucleus, what you see is that you don't just have these two droplets, we'll call them, that assemble. That's only sort of at the very end of the process. During assembly, you get actually nucleation of stuff everywhere, and it's very dynamic, right? So it looks like you get initially droplets that form everywhere, but then somehow they resolve or coarsen into just two foci. So all of this, rich non-equilibrium assembly dynamics has not been considered at all in this previous simple model we did. So we're going to try to think about that. We started calling these extra-nucleolar droplets, the stuff that forms initially everywhere and then goes away. I think I used that term once more. Okay, so to try to understand something about what's happening during this dynamic assembly process, we've been doing more and more work with in vitro systems, which have given us some insights into molecular interactions that are governing this, and gives us sort of a base platform from which to try to spring into the cell, if you like, and test the models. And so what we found is that a number of the proteins, so the nucleolus has got a whole bunch of these disordered proteins, components, and when we purify these and look at them under some kind of near physiological conditions in terms of buffer concentration, et cetera, what we find is that they form these beautiful droplets. There's a couple movies, the two of them sitting on a surface and fusing. This is a protein called FIB1, which is a circular protein. This is GAR1. These are just two examples. Okay, so they form beautiful droplets in vitro. And so, why would they not be doing this in cells, or maybe there's aspects of this that are richer and more interesting in the cell we can try to understand. But in vitro, still focused in vitro, if we we've mapped out the phase diagram of these things as a function of salt, so we can quench into this two-phase region, the droplets nucleate and grow and porcine as a function of time. So if you go forward in time, right after 2 minutes after you quench, it looks something like this. You get many little droplets, and you go an hour and a half, whatever later, these things end up in a small number of larger droplets. Okay, so many of you know that the time evolution of the size of these things, the average size, is often described in terms of these power law growth rates. So this would be the average size of these droplets, the distribution, but the average size of the droplets grows at some power in time. And here it's written as t to the n, and this is some growth prefactor. So the one that most people know is n equals a third, right? This is the Lifshitz-Lyazov kind of thing, or diffusion limited Oswald-Brangwynne, we'll call it here, DOR. You also get a 1/3 power in a regime where the system's evolving just due to Brownian motion coalescence, where the droplets are just diffusing around. Not Oswald-Brangwynne, where the big guys are essentially eating the little guys, but just where they're bumping into each other and coalescing. So you end up with this t to 1/3 scaling of the average size in time. Okay, so again, this is all in vitro. This is kind of the baseline for what we're trying to look at in cells. So I look at this data in vitro, mean drop radius as a function of time. You see that it really does follow this 1/3 power law, pretty nicely. People get all worked up about how many logs you have and stuff, but I mean, it looks pretty good to me. And you fit it, it's kind of right there in 1/3. This is just two different experiments where we add RNA, and RNA sort of accelerates the growth, but you still get this 1/3 power. As far as we can tell, based on the parameters that we have, we try to estimate this growth prefactor. We're pretty sure that at least in this in vitro experiment, this is in this Brownian motion coalescence regime. So with our confidence intervals on what we think this prefactor is, it seems to fit a little bit better for this Brownian motion coalescence than the diffusion limited Oswald-Brangwynne. The 1/3 is the same in both cases, but the prefactor is different. This one's a little bit underestimates things. So we think that at least in vitro, it's very nicely matches simple theoretical expectations. So the reason that the growth rate is larger in the RNA case, where you have RNA around, you can see that if you look at the phase diagram. Again, this is in vitro phase diagram for one of the nucleoproteins we're focused on. This is fibrillary. Sorry, I didn't mention that. It was the last one I showed as well. We see similar things with the other ones actually. So as a function of salt, the way we typically do these experiments is you start at a high salt concentration, quench down, and nucleate droplets. But you can see that at physiological salt concentrations, 150, let's say, millimolar, the phase boundary is at a micromolar or two, and that's in the physiological range. But what I want to point out here is that RNA shifts this phase boundary. So when RNA is around, you can get droplets that nucleate and grow at even lower concentrations. So RNA seems to somehow be promoting the assembly of these droplets. And we think that's important for what's going on inside the cell. And I'll try to tell you a little bit about that now. Okay, so RNA is shifting this phase boundary. It's promoting droplet assembly. This, immediately becomes something you pay attention to, because the nucleolus, what it's doing is it's assembling around sites of nascent ribosomal RNA transcription. Okay, so RNA is being transcribed at a particular gene locus, and then all this stuff is condensing around it. Okay, and that's, in this movie, I'll show you again, the time dependence in the assembly. What's happening is you're getting droplets kind of nucleating everywhere, and then they end up only at those two sites. Those are the two sites where ribosomal RNA is being transcribed. And you can actually see that. We've done some of this oligo fish probes. Basically, this is the same protein as fibrillary that I showed you in the in vitro data. This is now in vivo fibrillary. Here's the ribosomal RNA probes that are seeing the the RNA at these transcriptional sites. And so they, these things co-localize. The big droplets that eventually will, beat out these little guys. The little guys where there's no RNA eventually go away, but these big guys, are stabilized by the RNA apparently. And so this makes sense based on what I showed you in vitro. And to sort of further reinforce that point, we can do this experiment. The beauty of the cell organ system, we can manipulate all these genes at will with RNA interference. So what we do here is we inhibit this RNA pol-1, so this RNA polymerase 1 that is doing the transcription, is making the RNA at that site. And we basically knock down one of the key initiation factors for that and ask what happens. So what happens is that as this process moves forward, this is a nucleus of one of these cells in the embryo. So up until this point, it looks identical to what would happen in the wild-type case. But in the wild-type case, what would have happened, which I just showed you, right, is that these droplets would eventually go away, and you just have the two at the transcriptionally active sites. But in this experiment where we've knocked down this transcription, you just have these droplets, and they just kind of hang out and evolve and bump into each other and fuse and stuff, and then eventually they go away once the nuclear envelope breaks down, and the cell proceeds to the next cell cycle. So these droplets do not require RNA transcripts to form at that specific site. But when the transcription is active at that site, then these guys basically, they lose the game, right? They're destabilized. Okay. So we've been trying to understand this in a more formal way, right? Based on what I've told you about RNA shifting phase diagram. If you have transcriptionally active sites, then that sort of makes sense that those would be the preferred sites. We'd like to try to formalize that. And so we've been doing some work in close collaboration with Nico Patai, who's also at Princeton. He's done work mostly in thinking about solid state phase transitions, and also done a fair amount in membranes. And so we're thinking about this from a formalism that many of you probably know. This is kind of like a base formalism for thinking about phase transitions in polymeric systems. Floyd-Huggins, free energy. Here I've got n equals 1, so this is really a regular solution kind of free energy landscape. But basically this is a free energy as a function of concentration phi. So you can think about phi as some volume fraction or concentration variable. The first two terms here are what we get from, this is mixing entropy. So basically you can do the lattice model, do the Boltzmann entropy calculation and so forth, and you end up with this. This is what the mixing entropy looks like. This one is the one that does, mixing entropy, of course, wants things mixed, and so this is going to fight phase separation. The interaction energy is what could potentially drive phase separation, depending on this parameter chi, which I already showed you on a graph in cartoon form, right? The y-axis of that preliminary phase diagram for the nucleolus. We're going to try to dig into this thing and try to understand a little bit better. So chi in this formalism is essentially tells you, I'll show you this next thing here. If you're thinking about a polymer sitting somewhere and it's next to a solvent molecule, a segment of the polymer next to a solvent molecule, or a segment of the polymer next to another segment of the polymer, then you can think about basically whether the system energetically likes homotypic interactions or heterotypic interactions. So the interaction energy of a segment of the polymer next to a solvent, or polymer next to polymer, or solvent next to solvent. So these guys, if this interaction energy is really high, then the system wants to phase separate. So when this chi parameter is really large, you're driven into a two-phase. You get this curvature of the free energy landscape, you have this two-phase region. So this is kind of classic stuff in polymer physics as a baseline. This is what we've been thinking about. So the idea is that RNA promoting this phase separation, what it seems to be doing is effectively, you can treat it as impacting this chi parameter. Right? It's essentially making phase separation more favorable. And we're trying to model that through this chi parameter. And so the dynamics, again, this is something that people have probably often seen, the dynamics of phase separation. Often the description is taken as a Kahn-Hilliard model, So you see that this is a type of diffusion equation, right? This looks like a diffusion equation if this were concentration, right? So this is velocity of something here. This is basically an effective chemical potential, because the diffusion equation essentially reflects chemical potential, which is more or less the same as concentration when it's just entropy. So you've got this free energy. The free energy of the system is a function of this concentration variable. This is a square gradient free energy term, which penalizes sharp interfaces. So if you take the Flory-Huggins free energy landscape, you plug it in here, you end up with something like this. And so our collaborators got these beautiful spectral methods and so forth to simulate this. And so what we've been doing is trying to again, capture and see what the predictions that come out of that are. The idea that at the transcriptionally active site, RNA promotes condensation of this material through, essentially through this chi parameter. So we quench into this two-phase region of the whole system, and then we basically define a region where chi is larger, so there's a stronger driving force for phase separation. And this is the kind of simulation data that you get out. This is one of the regimes I'm going to show you. We've looked at a bunch of them. So you can see, you ask whether this is going to be something that looks like Brownian immersion coalescence dominated or diffusion limited osmolar ripening like you saw in vitro. There are these two possibilities. So this is a simulation. Again, so initially you nucleate these droplets everywhere, but the chi parameter is slightly larger at this model transcriptionally active site. This NOR chi parameter is slightly larger. And what that does is it basically outcompetes the droplets. So these droplets nucleate everywhere, just like in the experiments. But then because you've got this transcriptionally active site and RNA shifts the phase boundary through this chi, then you basically have one droplet that wins out. Okay, so we've been looking at these simulations and trying to see, compared to the experimental data and what the predictions and so forth. So you can see that you start out, it looks like you've got this diffusion limited growth as n to 1/2 regime at the beginning of these simulations, and then you cross into something that looks more like a n to the 1/3 number. N here is the time power in the average growth. So this is plotted slightly funny, plotted as volume, but that's the n to the 1/3 regime. Okay, and then what happens here is that as the system evolves, you eventually the nucleolus at the transcriptional site takes off. It's essentially this accelerated growth because all the other droplets basically dump their material into the nucleoplasm and they're eaten up at this NOR site. This is basically exactly what we see in vivo. We see that there's something at least consistent with the 1/3 power law growth rate of all these droplets. Now, I'm not at all going to argue that this is like solid evidence for a third. I think I'll make that stronger in vitro, but this I'm not going to make stronger. It's consistent with the third at least. But then what happens is, this is for all these droplets, these N's included. And then once you get to the later stage of evolution, the nucleolus just takes off, and these guys essentially go away. Yeah, sorry? Time? Oh, that's time. Okay, so I'll wrap up now. This is a good time. So, okay, the prediction, one of the key predictions of this model is that if RNA is shifting the phase boundary, that we could change the concentration and get into a regime where we get nucleoli to assemble, but not these extra nucleolar droplets. And that's exactly what we see. We can tune the concentration, so we just crossed into this basically gap between the RNA phase, the plus RNA phase boundary and the minus RNA phase boundary, and we can get these droplets to form. If we go further, then we cross into the region where they cross the phase boundary for these extranuclear droplets as well. There's other predictions that seem to be matched pretty well. Basically, the size of the droplets, the total number of droplets, and the size grows with concentration in a way that seems to match pretty well with the predictions of the model, and I can tell you more about that afterwards if you like. Okay. Okay, so I'll skip this part. I just want to say, the reason why we're excited about this is I told you the nucleolus drives cell growth. That's what it's doing. It's making ribosomes that drive cell growth. We think this is cool because what this says is that cell size, by impacting concentration, in turn feeds back on nuclear assembly and function. And this is sort of a key aspect of the biology that may have relevance to cancer, for example. We've developed some technologies and microfluidic platforms to study worm growth when these things are assembling and cycling worms. So I'll tell you, there's just 3 take-home messages. I started out with this kind of picture, trying to think about link scales, and I told you about some of the ways we're thinking about how those links work through the phase transition picture and the connection to cell size and concentration. Take-home message number 1, RNA protein bodies represent liquid phase droplets of RNA and protein. I'll show you a bunch of examples of that. Phase transition seems to govern nuclear assembly, both whether it assembles and if so its size. And then RNA transcription can play an important role in droplet assembly and shifting the stage boundary and impacting the dynamics of assembly through the activity of transcription at a particular site. Okay? So I'd like to thank folks in my lab. Most of the work that I told you about today is done by a really talented postdoc, Steph Weber. I showed you some other things from Shani Elbaum, Leshvai, and Sean Gloria. We've got a very interactive group, and so I think we're really contributing one to another. I'd like to thank iPoLS for funding and career award, who's really believe in this work, and I really appreciate that, and other funding sources as well. Miko Tai and Joe Berry really spearheaded the theoretical aspects of this work. I'd also mention that Sue Myung is a CPLC member who's been a close collaborator in some work that he wants about. Thank you very much.
A couple of questions.
Certain species, but also exclude others?
Yeah, that's a really great question. I think that's a key question in this field. How do you get specificity, especially when you think about these intrinsically disordered domains and the sequence motifs and the charge distribution and so forth. There's some recent work in our lab and other labs starting to tease this out, but it's not clear at all. You get coexistence of 2 different types of drops. Some of the components are similar between them, but some of them do not go in. What defines the partition coefficients, which would impact the activity. But there's really not much known at all about that, and it's a key question. Yeah.
So clearly you showed the relation between cell size and nucleolus size.
Yeah.
What about the ribosome biogenesis rate and cell growth rate in relation to these 2 size parameters?
Yeah, that's a great question as well. I mean, we're very interested in that, and this is sort of the stuff I skipped over. We've been using the embryo as a test tube, which has worked out well. We can manipulate these things to get at the biophysics. But then the question is, okay, is that happening in a growing worm, and how does that impact growth rates and stuff? So I just tell you, we're trying to do things like this. As the worm grows, this is just the intestine of the worm. This has one particular tissue and one particular cell type. But the cells get bigger and bigger as the worms get bigger and bigger, and the nucleoli get bigger and bigger. And so we're trying to quantify all this and see if it reflects the kind of phase equilibrium in the worm and how that impacts ribosome biogenesis, and then use these microfluidic platforms to look at the growth rates. I don't really have anything great to tell you other than that we're doing that. Yeah.
Liz, thank you for a very exciting talk.
Thank you for giving me time as well.